Hello, I'd like to welcome you to the Summer School webinar. My name's Alison McGregor and I'm the director of the programme. So first of all, before we actually start talking about what the programme is, I'd just like to do a few bits of very tedious, boring housekeeping with you. Now, I'm sure all of you are used to online meetings now. They seem to have been the norm for the past 18 months or so. But just to remind you, this is a Microsoft Teams session. We will be recording it so you can access and rerun the session if you wish to. And with that, there's the usual functionality. So if you want to post us some questions, you can use the chat line. And please also feel free to use any applause or thumbs up or any other words of encouragement for the team as they try and deliver this webinar to you all. So I suppose it's up to me to tell you why you should come and join our summer school. What's so exciting about it? And I know you might all be a little bit sick of this pandemic that's going on in the background in all of our lives, but our summer school is actually all about Imperial and the pandemic, because we as a university have played quite a key role. So if some of you have been a little bit frustrated perhaps, or not understanding where some of the progress has come or some of the guidelines have come, and you want to learn more about some of the modelling that Imperial's done to try and manage and predict the progression of the pandemic, the summer school is obviously the place for you. It's also going to give you highlights in some of the work done around the testing program of how we tried to look at actually whether the modelling was being borne out by the testing results, and the work we've done to make sure the work environment and public transport has been safe for people to use. So we have a huge range of our specialists speaking, including Wendy Barkley, Rodney Shattuck and Neil Ferguson, all of them in different ways playing a key role in managing this pandemic. So not only are you going to get to listen to them, you'll be able to ask some questions of them, and you'll learn some of the techniques that have been used. So I suppose then you say, well, okay, that's great. They're being interviewed by the newspapers and the media. Why do I need to speak to them? You're going to be hearing about them talking as scientists, passionate scientists, committed to their work, and you're going to be able to actually pose your questions, not the questions that the media think you want to answer. And I think their coverage will be much more diverse than you're used to from normal press reports. But there's much more to joining a summer school. It's a huge opportunity to engage with scientists, to get an insight whether science is a career or something that you want to learn to be passionate about. We love having students come to our summer school. It's usually a highlight of our year. And the fact that we're having to run it online is a slight disappointment to us, because normally it's such a fantastic interactive program where we get to explore London and we get to know all of you really, really well. But we're going to try our very best to do that online and make this a dynamic school and a chance to meet people and start your scientific networks already, both with staff here at Imperial who are going to support you through this journey, listening to our experts, and meeting people from around the globe who are also passionate and interested in a career in science. So we will answer some questions for you at the end of this session, and I'm joined on the webinar today by some amazing people, including Geoffrey Vernon, who is the academic lead for the summer school, and he's going to be telling you a little bit about what you can expect from the school. We've got the lovely Olivia and Steph, who are two of our academic session leads, and they'll be demonstrating how we're going to deliver some of the teaching to you during this webinar, during this summer school, I apologise. And then, of course, we've got Meg. Meg is just so key. She is our administrative lead and summer school guru. She is the person that makes sure you have fun. And actually, she makes sure we have fun, and she makes sure we become one big summer school family. So all I can say is you have the most fantastic team working on this summer school. It is an amazing opportunity. You'll get some unique insights into science, and we can't wait to welcome you to the summer school. Please feel free to ask questions at the end, and the team will do their very, very best to answer them. And I hope to see you later in the year. Take good care, everyone. Bye for now.
Thanks very much, Alison. I'm Geoffrey Vernon. I'm the academic coordinator for the summer school. And what that means is that I have planned the content of the school, and I've recruited the academics who will be working with you. So anything that's good or bad about the school, you can blame me for it. We've been running the summer school now since 2015. It's called Revolutions in Biomedicine. And we always use the summer school to showcase what we're doing at Imperial. We have a fantastic range of biomedical research at Imperial. We train doctors, we train basic scientists, and we like to give the students who come to the school a flavour of what we do here. We want you to feel what it would be like to work in medicine or biomedicine, and maybe some of you will think of coming here to do graduate work with us, and some of our summer school alumni come back and we see them again. If this was a normal year, you'd come to our campus in London and we'd all be meeting in person. In 2020, we reluctantly cancelled the summer school, but this year we thought we're not going to have a second year with no summer school, and we've decided to go for it. So welcome to Revolutions in Biomedicine Online. It occurs to me that that spells ribo, and if you're interested in ribosomes or RNA, that will mean something to you. Our theme this year is Pandemic Imperial, and the sub-theme is called Populations, Patients and Point Mutations. And we picked that name not just because it has 3 P's in it, but because it captures everything that we do at Imperial in response to COVID. So the population dimension is the environment, the context where disease happens and where epidemics happen and where we can model them. The patients, that's the individuals and their physiology and their disease state and their treatment. And the point mutation sub-theme is about everything that goes on that we can't see, below the surface of the cell. It's what happens to the pathways in the cells, the molecules, the proteins, the genes. And of course, it's also what happens to the virus and the way it interacts with our cells and makes us sick. So I ought to tell you how we're approaching all of that this year. And we know that everyone is fed up with learning through a screen. You've probably spent much of the year staring at people talking at you. And we're all fed up with teaching through a screen as well. I sometimes have to remind myself to think like a radio chat show host. I feel as if I'm broadcasting to the airwaves, not really sure if anyone's listened to me or not. And I might encourage the audience to ring up and phone if they think they have anything they want to comment on. And often when I'm teaching, that's the kind of feeling I have. I'm not sure if anyone's out there. We want to overcome barrier that the screen puts in the way. And we've thought quite a lot about the design of the sessions that we're doing this year. We want to make the summer school this year both authentic and interactive. Well, that's an easy thing to say, but what do I mean by that? It's going to be authentic because we want to use the kinds of approaches and tools and data that we actually use in science. So we want you, when you're on the summer school, to feel what it's like to do modeling in epidemiology, or what it's like to use patient data to understand infectious disease processes, or to take what we know about virus structure to design a vaccine based on it. We're always talking about whether the vaccines will be resistant to new strains that are coming along. One of the things we'll get you to think about during the summer school is emergent strains and how the vaccines could be designed based on those, and also the technology of vaccine production. These are just a few examples. You can read about the whole timetable on our website. But we have 15 different topics throughout the 3 weeks where you will see the frontiers of COVID research as we're doing it at Imperial, and you will have a chance to interact with our academics and with each other to explore those themes. So I said authentic and interactive, and now I'm talking about how you'll be interacting. We will get you to work on actual data that we use, or tools that we use in our science, or ways of thinking that we have to apply when we're working with COVID. We're doing it this way, partly it's because it's the way that scientists and professionals work. Sometimes at a conference, we do what you're doing now. You're looking at somebody talking, they may be showing their slides, they may be explaining their data. We do that some of the time, but it's not what we do all day. It's not natural to stare at somebody talking for hours on end and hoping to learn something that way. The rest of the day, when we're not watching somebody at a conference. We're working with our data, we're writing grants, we're producing a poster, we're designing an experiment, we're making recommendations to the health authorities. We're wondering about problems that the rest of the world haven't really thought about yet. You know, one of these emerging things that's coming up is long COVID. I don't know if you've heard of long COVID, but you know, it's the idea that people, after they have the acute infection, might then have a long-lasting disease that doesn't really seem to be clearing up, and it persists for weeks, months, might even persist for years. And long COVID is one of these new emerging themes that we'll be thinking about in the summer school. We've got 2 sessions dedicated to it. So we want you to get a taste of all that and how exciting it is. But the second reason we've designed it as an interactive summer school is because that's the best way to learn something new and challenging, right? There are some of us who learn well by being told something once. If I told you 10 cool things about the brain, you'd probably forget 9 of them. If you wanted to remember all of them, I would have to find some kind of challenge or scenario or problem for you to think about so that you could respond to that material and do something new with it. Most of us have to learn like that. We have to work with what we've heard or a problem that's been set, and then produce an output, and then discuss that with other people. And then sometimes we find that our idea was not a good one, or it might be a good idea, but there's a better idea in the room. So you'll get a flavour of working like that in the summer school. To learn something new so that it sticks, you have to play with that idea, apply it to a new situation, work on a challenge that might not have a single answer. There might be alternate answers. As well as thinking about how you'll learn on the school, we've thought about how you can be assessed. So the assessment for our summer school is optional, but it's a great challenge if you want to get involved with it. So we've done away with the exam this year. What used to happen is on the last day of the summer school or the day before, we got you all into an exam hall, we logged you online, you did an online exam, and then we marked that. This year we're doing something different. We want you to assemble a portfolio of what you're learning during the summer school, and then you'll have the chance to select and submit the best bits of your portfolio after the summer school ends. So there'll be an interval after the summer school where you can finish working on the portfolio before you submit it and before we assess it. The portfolio is a document of what you've learned and what you've enjoyed learning and what you've explored, not an exam paper that you forget about the next day. I mean, think about it. You do an exam and no one even gives you the paper. You're not allowed to take it away. The portfolio isn't like that. So I've talked enough about it. I hope to see you all at the summer school. I'm going to hand you over to my colleague, Steph Askoff. She's one of the instructors on the course. And I think you'll agree when you've heard her that the lab she's working in is doing something rather unique, which is one of the things we want to share with you. And you will have a chance at the summer school to work on the kind of questions that Steph thinks about all day. So over to you, Steph.
Hi, I'm Stephanie Askoff, and I'm going to talk a little bit about our research in the Department of Infectious Diseases at Imperial College and how we're going to explore this in the summer school session that we have planned. So, in the Department of Infectious Diseases, we use clinical samples to look at immunity to common respiratory viral infections. These include pandemic influenza, RSV, and COVID-19. So we do this using controlled human infection studies. These involve deliberately exposing healthy volunteers to infectious organisms. These can include viruses, bacteria, and parasites. So why would researchers want to carry out such studies? They provide us with vital data that tell us how these infectious organisms cause disease and how they spread and how the diseases can be prevented or treated. Within the field of human challenge studies, There have been a range of pathogens which have been used, and volunteers have been infected with rotavirus, whooping cough, pneumococcus, influenza, typhoid, malaria, or RSV. So why do we carry out this research? So controlled human infections have some major advantages over other studies we might perform. Most notably, the infections are actually in the human host, not a laboratory model, such as a mouse or rat. And unlike observational studies of community-acquired infections, we can infect the participants with a known amount of the pathogen at a set time, and this allows us to study the very early time points in infection. And we can also pre-select our participants so that we avoid or even potentially include individuals with pre-existing illnesses such as COPD or asthma. And these illnesses may affect how they respond to the pathogen. The high infection rate associated with experimental challenge studies allows us to draw conclusions from much smaller sample sizes than studies of naturally occurring infections, which may require hundreds or even thousands of participants to reach a statistically significant conclusion. We can carry out intensive samplings of multiple compartments, such as blood, tissues, or mucosal samples. And we can also follow the participants over the course of several months or even years to determine how infection resolves and long-term immunity develops. But there are also disadvantages to these studies, and these include the expensive ethics and safeties required to carry out such studies, the expense and the complexity involved, and there are a limited number of challenge strains of the pathogens which have been lab grown to the precise standards required. And these strains may differ from naturally occurring strains over time, especially in the case of viruses that mutate very quickly, such as influenza. Finally, we can only induce mild to moderate disease in participants, so it's important to bear this in mind when we're trying to extrapolate to severe forms of the disease, especially in high-risk individuals. And these difficulties are why there are only a handful of institutions in the world capable of carrying out these studies, of which Imperial College is one. So, currently, our group is involved in the world's first human challenge studies infecting healthy volunteers with the SARS-CoV-2 virus. And these challenges will be used to study adaptive and innate immune responses to the infection. And eventually, we hope that they will be able to help us establish quickly which novel vaccines are most likely to succeed. And how do we carry out this research? So as we're infecting our volunteers with human pathogens, we need to keep them quarantined in our clinical research facility. This offers a managed setting with healthcare support for the participants, and it also acts to prevent the spread of the disease within the wider community. The quarantine period allows us to intensively sample the participants, taking blood and airway samples over the time they're quarantined with us. And participants return for follow-up samples at several weeks to months post-infection, allowing us to investigate long-term immune responses to viral infections. All post-infection samples can be compared to pre-infection baseline samples, giving us a unique data set to work with. So a human challenge model can help researchers answer a lot of questions about a disease. These questions include what's different about the immune system of people who get infected, and why do some people not catch the same infection, or why do they show no symptoms? Why do some people shed a lot of virus? And how does the virus spread and adapt to hosts? And importantly, how long does natural immunity last? And what factors put people at higher risk of reinfection? So this is the first time that a human challenge study has been carried out during the middle of a pandemic in this way. And in addition to the normal ethical and practical considerations that we face doing our challenge studies, the COVID-19 infection study has had to face unprecedented challenges, such as the emergence of new variants, a fast-changing treatment and vaccine landscape, and new immunological questions surrounding phenomena like long COVID. These are real problems we need to solve. And in the summer school, you'll get a chance to work on current questions like this in my session. And we're going to come up with solutions we can share and discuss. You'll be asked to put yourself in the position of a researcher designing a reactive COVID challenge study capable of responding to real-world pandemic conditions. And hopefully we can come up with some answers together. So thank you for listening. And if you want to find out about human infection challenge studies in the meantime, especially the COVID challenge studies, here are some useful resources that I can guide you to for further reading. Thank you, and I hope to see you next month.
Hi there. So my name is Olivia Boyd, and I will be discussing with you today one of the summer school sessions that will be available to you within the school, titled Real-Time Monitoring of Emerging Variants. And this session is conducted by myself, as well as one of my colleagues, Fabrizia Ferreira do Nascimento, who is a postdoc in my research group with me. I'm a PhD student there, and we work with Eric Voltz. And essentially what we do in our group is we combine genetic and epidemiological methods, which is called phylogenetic methodology, to look at the COVID pandemic outbreak. And so just to give an idea of the overall session and what we will cover, in Fabrizia's portion, we will look at phylogenetics, including what is a phylogeny, why do we use phylogenetic trees in research, which we'll learn about in the session. As well as phylodynamic methods and epidemiology. And here specifically we'll focus on the basic concepts on compartmental models in epidemiology, as well as how we apply phylogenetics and phylodynamic methods in research. And for my portion of the session, we'll then look at mutations, vaccines, and vaccine escape. And specifically here, we'll identify how mutations occur in RNA viruses with a focus on evolution, as well as why mutations are important for vaccine effectiveness and understanding vaccine escape. And next, we'll look at emerging variants and why these are important to monitor in real time and what methods we use for identifying variants of interest or concern. And this is the portion of the session I'll be walking you through today. And so what do we mean by variants? Essentially, what we mean here is a lineage defined by epidemiological, geographical, and evolutionary characteristics of interest. And it's important to note that, for example, with this new WHO nomenclature, we have here our alpha variant, or the B117 lineage, which appeared last fall and became the dominant lineage in the UK. But what's important is we can also have a variant that includes the lineage with a specific mutation of interest. For example, here we have this additional mutation that might have some immune escape that we should be worried about. Why are these important to identify? Because they can help us predict what's going on in the outbreak. More specifically, different variants have different transmission severity or immune escape. So it's important to monitor these and understand what's happening when we have emergent variants that appear in the population. So how do we go about monitoring these variants? First, we have to consider what outbreak information do we have. So as part of the pandemic response, the COG UK consortium was created, the COVID-19 Genomics UK consortium. And essentially what they do is they sequence viral samples from cases in the UK, and they try to sequence as many samples as possible to have an understanding of the variant variability in the population. And all of these sequences either come from hospital samples or community samples. And as a small side note, the importance of being a hospital or community, also known as Pillar 1 or Pillar 2 samples, we have to consider community samples are representative of the underlying outbreak in the general population as a whole. Whereas hospital samples are more likely to represent more severe cases and less likely to be representative of what's circulating in the actual general population. And so something for you to think about, and something we'll cover in the session as well, is when would we use these specific sample sets. The other information we have is epidemiological data from Public Health England that we can link back to our viral sequence data. And this includes things like age, gender, vaccine status, sample date, severity of the case, pillar being the hospital or community sample type, and then outer postcode as well. So we have geographical location attached to our sequences. So how do we integrate these together? We develop an outbreak scanning tool. And essentially what this is a combination of multiple phylogenetic methods that we use to monitor the UK pandemic in real time. And so with this work that you'll be actually using yourselves within the session is work that we developed as part of the SARS-CoV-2 UK pandemic response. And when we think about monitoring emerging strains, we have to consider what we're interested in identifying actually when we're trying to identify whether there's a variant or strain of concern. So the first thing we can look at is highest growth rate clusters. And so we can use things like logistic regression, for example, to compare clusters in our population. And what we mean by a cluster here is essentially a group of cases that are linked genetically and epidemiologically, either in terms of geographical location or through contact tracing, things like this. And so, for example, here we can see this being our newly named alpha variant or the B117 variant, also colloquially called the Kent variant. Earlier on in the pandemic was dominating the high growth rates, but now as we see that our Delta variant has come out, this B16172, this is now actually dominating the high growth rate clusters, on our y-axis being the growth rate itself. The x-axis just represents here essentially a log ranking in terms of highest growth rate cluster to lowest. And here what I've shown to compare is also the other lineages that are popping up in the population, which you can see there are many, many others that we include here. We can also look at these growth rates over time. And so what we have here on the x-axis is our last possible sample date. And on the y-axis, again, our logistic growth rate. And what this means here, when I say last possible sample date, it means we include only sequences up to that sample date. And we can clearly see here, this is the Delta variant again, that it's taking off as we include more and more case data over time. And actually, this is a bit outdated now. Now, as we move into further in June, and we have data coming in daily that we can update this with, we find that this particular lineage or variant is taking off quite a bit now. Next, we can also look at specifically the frequency of the variants of interest. So this is, again, our Delta variant. And to give you an example, this is a recent output where we see, okay, this Delta variant is actually representing 60% or 0.6 out of 1 of the sequences in the population or the cases in the population. And this is actually, again, slightly outdated. We actually now see it's about 95%. But using the methods we've developed, we can monitor this. We can also monitor, again, comparing the growth rate of this variant compared to others using geographically matched samples. And as well, we can look at specific regional clusters or local tier level authorities in the UK. And we see that this is really taking off. It's in over 200 locations now. Next, we also look at large shifts in mutation. So what you see here is a phylogenetic tree. And what we are, for example, trying to see is if we have a cluster that comes as a standout, meaning it has quite a large genetic distance from the rest of the sequences within the tree. And so, for example, when our alpha or B117 variant first appeared in the population in the fall last year, we saw that this actually had a large genetic distance from the rest of the tree, which suggests that there was a large shift in this particular lineage. And why is this important to monitor? Because we are concerned about immune escape or vaccine escape, and this is where this is likely to occur. We can also look at mutations of interest. So, for example, looking within our specific alpha variant or B117, in February, we had a cluster of concern in Newcastle, up in the north of England, that had a mutation of interest, this Q677H. Which is a spike protein mutation, and essentially it helps the virus bind to our cells better. And so what we wanted to monitor is if this cluster was actually spreading beyond Newcastle and growing. And actually what we found as we were tracking it is that the cases were reducing over time, so it became less of a concern. But what we can also look at is other mutations linked within this cluster. And so, for example, we'll go through a list of mutations within the session itself. But just to give an idea that when we identify one mutation of interest, we can also see if there are other mutations of interest that occur with this mutation that might be causing higher transmissibility within a lineage of interest. We can also look at age distributions within our clusters. And this is particularly important as we move forward with more vaccine efforts, because now we find we have pockets of age groups that are less protected against the outbreak because they have lower number of vaccinations occurring. And then finally, most importantly, moving forward, we can look at vaccine escape. So we combine all these things together to assess essentially whether we're seeing vaccine escape occurring in the population and what this means for future vaccine development and the future of the outbreak moving forward. And so the important thing that I think you will take away from this session as well is that, you know, we have hundreds of lineages. Oh, uh-oh, something happened. Sorry, I think someone took control of my lecture slides. Hang on a second. Here we go. Sorry about that. So basically what we can do is we can use the scanning tool to look at these hundreds of lineages. And when you think about for each lineages, there's also lots of mutations that are potentially linked to that lineage. So that's an exponential amount of things that we really need to look at when we're monitoring the outbreak. And here we just have a selection of some lineages of interest and the cumulative samples on a log scale over time. So you can see when they're emerging on this time scale. And essentially what we're monitoring is, are they really taking off? So for example, here we can see the Delta variant is actually becoming quite prevalent in the population. So this is something of concern we need to keep tracking. And so finally, we come to what we'll actually be using in the session. So here we have a scanning tool that we've developed. And what it does is it uses real-time data that we get in daily to monitor the outbreak. And so what we input into the scanning tool itself is a phylogenetic tree, which you'll learn about how to make in the session, and associated metadata that I mentioned before. And what we do is we incorporate multiple statistical methods into this package to assess selected clusters of interest or concern. And this includes looking at the evolution of the clusters, the growth over time and space, age distributions, and immune escape. And finally, what this does is it combines all this information in one place, where then you as the students can select a lineage and mutation, or cutoff of interest. Cutoff meaning, for example, perhaps you want to look at the top 10 highest growth clusters. And what's been really nice with the scanning tool is actually, in our group, we further developed this into systematic reporting for Public Health England. But what you'll be using this session is an online open source platform that we've developed for the public to use. And so within the session itself, we will have a practical portion. You will have a pre-recorded tutorial available prior to the session to learn about the scanning tool and how to use it. We'll go through the summary of this in the lecture, as well as the methods involved, and sort of a brief summary of what these methods are and how we implement them, as well as, of course, a Q&A portion so that you can make sure you understand what's going on. And then in terms of practical group work, you as the students will actually work together to develop a situation report for Public Health England, looking at specific variants of concern or interest, as well as the highest growth clusters in the UK. And this will be part of the summer school portfolio that Jeffrey mentioned before. And I think what's really nice here is you'll have a chance to actually create a report that's very similar to how we've been reporting throughout the pandemic. And you'll be able to create your own format for this if you like, but we will give you an example report as well. And the figures and tables are downloadable, so they'll be easily pasteable into your report format. And questions we'll consider throughout the session, for example, are where are the highest growth rate clusters? Are we observing immune escape in the population? Do we have some genetic shift occurring? Are there mutations of concern within these clusters? And as well, looking at age distributions in the high growth clusters. And of course, as I mentioned earlier, a list of mutations of concern will be provided, so you won't be expected to learn all of the mutations of interest that are existing out there. And I think this summarizes the session so far. And yeah, I guess now we move to a Q&A session where you can ask any questions you like. Thank you.
Thank you, Olivia. And listening to those talks by Olivia and Steph, I was just struck by how COVID seems to, it captures all of science. So whether you're interested in mathematical modeling at one level, to the itty bitty question of the virus and its structure at the other level, to how genetic mutations acquired, or what kinds of disease symptoms people have, and how we can use things like deliberate infection to try and understand the really early phases of the disease. You know, I think there's something for everyone in the COVID theme. And I say that because when Alison, our wonderful director, first proposed having Pandemic Imperial as the theme, I had my doubts. I thought, won't everyone have got sick of COVID by the time, you know, not sick in the literal sense, though some people did, but won't everyone have got tired of the theme? It just seems to be everywhere. But the brilliance of the idea that Alison had is that it really does cover all levels of organisation. So that's why we want in the summer school to have this portfolio where you pick the things that you're interested in, and choose them to investigate, you know, with guidance from us. So we do have some questions. Keep them coming in. If you don't, you should be able to find the Q&A tool at the top of your bar, and we can still take questions. But the first one that's come in is a very good question. It's, how will this program aid my career progression in biomedical science? Now, I take it that the student who asked that is a student of biomedical science, and they're going into that as a profession. So they might be working in a hospital pathology lab or something like that. I would say whatever career you're choosing, so whether you're going to be a practitioner, whether you're going to be a researcher, I think the great thing about the summer school is precisely this synoptic overview it gives you of across science. So if you take a theme like COVID, the summer school explores that in different ways, and it gives you the chance to put yourself in the position of the researcher or the practitioner. And I think this is a great growth opportunity because you get, in a way, hands-on experience of what it feels like to do that work. If you're then filling in a CV or a job application, or you're going for an interview, you've got things to talk about that you might not have been able to talk about if you just did your course. You know, you've now been on this experience where you've had to think about scenarios in biomedicine. And, you know, speaking of someone who's had to kind of recruit people into a research group, I look for those people who show curiosity beyond their science. So the people who've done a really brilliant degree, of course, I'm interested in them, but at the same time, I'm interested in people who bring a bit of value added, you know, the ones who've thought about problems, have thought about ways of solving them. They've networked with other people to discuss different solutions to things. So the summer school is meant to provide a kind of occasion to do that kind of thing. I will say, just putting it on your CV would look pretty good if you said, you know, you'd gone to the summer school at Imperial, but I wouldn't be impressed by that alone. If I was reading your CV, I'd want you to say what you learned by coming to it. And that's the point of the portfolio, because that's where you have to think, well, actually, what have I learned? So that's a great question. If that didn't really answer what you wanted, please ask another one in the chat. Another question's come in asking, can I acquire some knowledge about the present clinical trials? This might refer to Steph's talk. So Steph was talking about the REACT study where we deliberately infect healthy volunteers. Steph has said that if anyone wants to ask questions about that, she's happy to email you afterwards. But I'll tell you the very basic parts of it I know, which is that the trial starts off with 90 healthy volunteers aged between 18 and 30, because they're the ones less likely to have serious side effects. And they're going to be deliberately infected with controlled doses of laboratory strains. And the idea of that is to see what happens at the very earliest stages of the disease. Because if you think about it, by the time people go to the hospital, or they go to the doctor, or they start taking a test like a PCR test, they may already have symptoms. They might have been infected for some days or some weeks. So the idea of these early challenge studies is to see what happens, you know, in the hours and, well, really the few hours after infection. Where's the virus going? What receptors is it docking onto? You know, what's the time course of the disease? There's a third question here. So there's a question: will any of the content be assessed, and will it be graded? I've talked a bit about that. So I've said we haven't got the exam this year. If you're coming on the summer school, we've got a portfolio assessment. So that's where you, negotiating with our tutors, decide how you want to showcase your learning, right? How can you present the things that you've been most fascinated by and that you've had a chance to investigate and explore, how can you put that together in a portfolio and submit it after the summer school ends? It's optional, you don't have to do that, but I think students who take on that challenge of compiling the portfolio will get most benefit out of it. And I think particularly when you're learning online, it's easy to lose track of your learning. So we've introduced the portfolio deliberately so that you keep some kind of journal, because you might watch a really cool seminar or take part in a group activity on day one. Unless you're writing something down, responding in some way, or doing a task based on that, you probably forget on day two what you did. So the idea of the portfolio is to keep you, if you like, tracking your own learning. I've got a question which Alison, our summer school director, might like to pick up, because Alison's a professor in surgery and cancer. It's a question that doesn't really fit the theme of the summer school, but I'll throw it to Alison, and you can take it any way you like, Alison. It's nowadays, what methods can be used to treat terminal cancer patients?
Thanks, Jeffrey. And yeah, you're absolutely right. It's actually not the theme of the summer school. And I think it's a question that doesn't have a simple answer. It's like all things in medicine, there's never quite one answer. So cancer takes many shapes or forms, and it depends what sort of cancer is, how you treat it and how you respond. And also it depends a little bit on how you define end-stage cancer. So there are lots of approaches. There's surgery, there's chemotherapy, there's radiotherapy. It really does vary with the different types of cancer, the age of the person, and all their other comorbidities. So there's not quite a simple answer. And I guess maybe what some of this course will help you do if you join it is work out how you can find out that answer yourself. So sometimes you don't always, I'm hoping at the end of this course you won't need somebody like me to answer that question. You'll feel a lot more confident to go into the literature and find the specific answers that you need. And I guess one of the early questions that was posed is what will I get out of it? What's the benefits of doing this course? So it's taking your portfolio of skills up another level, but this is also your chance to start forming your own scientific network. One of the things about a summer school is, okay, normally it's in person, but even online you will meet people from all around the world who come to join the summer school, plus all the different staff lecturing in. Actually, they form your research network, and they will form people that you can go to for advice or help develop questions or ideas with. So, that's one of the other reasons for engaging with something like this, is it gives you the skills to answer questions like the one you've just posed me, but also it gives you the network of people that you can reach out to throughout your research careers. And although the world's very big, science is incredibly small sometimes. And I'm doing a project with engineering at the moment, and one of my colleagues there said, oh, I've got this contact interested in our project, wanted to know the code for this, that, and the next thing. And I said, oh yeah, I know them. They could have just come to me directly. And it's like small world, they might be on the other side of the world, but you already know them. So this is a chance to make new friends and form your own scientific network. So sorry to go slightly off piece, Jeffrey, but you know what I'm like.
I do, and I think I have the same tendency. So divergent minds think alike, or perhaps differently. I don't know. One of the questions that's come in is, how in-depth should our knowledge of essential cell biology be? So essential cell biology, the Albertson Bray book, is recommended reading if you want to get up to speed. My answer to that is, you should be comfortable with the concepts that a first-year bioscientist should be comfortable with. So what's a cell, what's a molecule, what's a membrane, all those. It's not required that you know a lot of stuff before you come. We're really looking for approaches and attitudes. We're looking for people who are open to thinking about challenges, working with other students, and interacting with staff to put themselves in the position of researchers. So I would never say that knowledge for its own sake is pointless. If you happen to know a lot of stuff about cells, molecules, genes, protein folding, mathematical modeling, epidemiology, that's all great. That will help you. But the main thing we're looking for is people who want to come in the spirit of the summer school, to try some of these exploratory things, and perhaps to try some things that are out of their comfort zone. If you fear the idea of mathematical modeling, this is a chance to get friendly with it, because in COVID we talk about it a lot, and if you're the kind of person who thinks, I wish I knew what they were talking about when they're saying, here's the prediction for the number of cases in the autumn, then the summer school's a place, so a safe space to do, if you like, unfamiliar things that you don't quite feel comfortable with. A question has come in about social activities, and I think I'm going to hand you over to Meg for this. So the question is, will there be any social activities during the online summer school? And will we get a chance to meet any Imperial students?
So yes, there will be social activities. These will happen outside of your timetable of teaching, and they will be sessions that are facilitated by 2 student ambassadors who are current master's students with us, so you'll get to meet the Imperial students that way as well. We will also have a Ask Us Anything channel. So this is really your place so you can question our student ambassadors and find out more about life at Imperial, and you'll get to hear the students' perspective. In addition to that, there'll also be some further chances to interact with some students in a careers session where you'll also meet some of our current PhD students.
Thank you, Meg. Yeah, we're very keen that you should know kind of what it's like to be a student here, and there will be plenty of chances to talk to our students in different guises, some of them as mentors, some of them as ambassadors for the course. There's been a question on technology. So besides MS Teams, what other software do we need to use during the summer school? Now, when I was watching Olivia's session and also Steph's, I thought, okay, there will be packages and tools that you need to use while you're in the session, but all of that will be contained inside the session. I think that's right, Olivia. You don't require anyone to own a package separately or to download anything other than that.
Yeah, that's right. It's an online, it's just an online tool, so it's publicly available. So it'll be very easy and accessible for the students to get to.
Yeah, so we've deliberately chosen technology platforms that we know students can use anywhere in the world, because we do have international students. We don't want to pick a tool that, I don't know, you can't open in New Zealand or that doesn't work in Peru, for example. So we know, partly because we're a very international campus, and we have international courses like MBAs that recruit students all over the world. We know that MS Teams is a fairly reliable and stable platform for delivering a course. We've just got to find interactive and engaging ways of making that work for you and your learning. There's been a question about how the portfolio will be marked. That is something we will be guiding you about that at the beginning of the school. So the question about whether the grades go towards anything, that's really between you and your institution. So if your institution says you can do credit on a short course and that you can count that as part of your degree, that is something to negotiate with them. But from us, you will get a diploma saying that you've taken part, you've done your portfolio, this was how it was graded. But how that counts towards your own studies is something for you to discuss with your teaching team. But the kinds of things we'll be looking for is the evidence that you've learned something, the evidence that you've gone through a learning journey, so you haven't just compiled a portfolio of things you knew already, and how you've reflected on that. Now you've gone through that experience, what would you do next time you met something unfamiliar and challenging? Do you think you would be able to rise to that challenge? That's going to be an important aspect of the portfolio. There's been a question on entry requirements for the course. Is that something that you want to take, Meg?
Of course. So for the entry requirements, I'll post a link in the chat because it's all listed on our website, but you will need to have passed at least one undergraduate biosciences module. You will also need to meet a certain level of English language, which is all listed out very clearly on the website, so that's the best place to check it out. And you will need to be at least 18 years or over.
Thank you, Meg. There's been a question about talking to the other summer school participants outside of the sessions. I think that comes back to the social activities that Meg was talking about. we do want you all to be working with each other. The summer school will have failed if at the end of it you say, well, I didn't really make any contacts, or I didn't make any friends, or I never found myself working in a group with people that I could, you know, address a problem with. So the whole point of the summer school is being able to, you know, make connections with people you wouldn't otherwise have met. So definitely there will be opportunities to do that. We've been asked if there'll be interactive breakout sessions every day. I can't promise every day, but I know for sure that sessions like Olivia's and Steph's will both involve breakout sessions. And there's going to be a mixture of those. Some of them they'll be strongly scaffolded, as we say. So you'll be told what the task is, what the expected output is, what time we want you to complete that by. There'll be other things that are a bit more open-ended. You know, the problem might not be very well defined, and you and your team might have to decide how to address that problem and what kind of output to produce. You might have some freedom of choice over how to do that. But if there was one word about the summer school I would like people to think of, it's interactivity. It won't work if it's not going to be a school where you watch the screen and listen to people giving you long lectures without tasks or opportunities to respond. And I can see that Nosheen has posted the entry requirements in the chat, so that's something you can click on if you want to follow that link. We have actually come to time, so unless Meg or Nosheen can point out a question I haven't taken or something I haven't said, Meg.
The only one that I don't think's been covered yet is how many students will be at this year's summer school.
Oh.
Do you want to take that one, Jeffrey?
Let's tag team. Meg, you go first.
So we currently have 30 students who will be joining us this year. Our hope is for a few more, but we'll keep the cohort to a nice size where you'll be able to work very collaboratively with each other and get to know each other very well. Do you have anything you want to add to that, Jeffrey?
No, I mean, it's very much the point. There are entry requirements. We are looking for people we want to pick people who we think will benefit from the course and who write in their applications things that make us think this will help them. It won't work if we've got 100 people. It won't be a good interactive experience for you. So yeah, we are going to close registrations at a certain point. And as far as I can see, that is all of the questions answered, but this isn't the end. So if you have other questions, please do post them to us and we'll do our best to reply. We are closing registrations towards the end of June. Is it the 28th of June, the last date? So if you are thinking of coming, please put your applications in soon because we need to set you up on our system. We need to give you your joining instructions. It won't work if you try and join at the last minute. I hope this webinar has been informative for you. I'm not just saying this, I have learned things by listening to all of the other contributors. And if you do have any more questions, let us know. But I hope very much that we'll be seeing you on the summer school when it begins in mid-July. Thank you all very much.